next speaker is Dr. Rezaei. Dr. Rezaei is Iranian, from Iranian nationality, but he works at INRA Julian-Jossas in Ile-de-France, that is Parisian region. He did his PhD thesis in the Museum of Natural History in Paris, and his specialty is biochemist and biophysics. He studies basically conformational dynamics of ovine prion proteins. He was my boss at my postdoc position there in France, and also it's a huge collaborator here with Dr. Coralia Rivas and Dr. Gustavo Moraga, also with myself. So I introduce you to Uman. I let you with him.
So thank you very much. So I will do quick. So I know that everybody's are hungry. So first of all, I would like to thank the organizer, especially Carola. It's a very interesting meeting because during these days we mixed neuroscience, neurogenerations, sexual behavior. So it's quite a nice mix of all of this. So today I'm going to speak about the concept of prion pathology, or all the concept of the prions, and more specifically the transmissible encephalopathy spongiform. So to start, few words about prion pathology. So prion pathology is a neurodegenerative disease. It touches much more motor and sensitive dysfunctions. It's induced neuron loss and spongios and astrocytosis. As here you can see here you have normal tissue, and when you have prion pathology going on, you have loss of neurons as you have formation of vacuole. That means that you lose neurons in fact. And you have a astrocytosis that is hallmark of inflammatory in the brain. What's very important in prion disease that you have an accumulation of misfolded conformation of PRP proteins. I will come on this point a little bit later. And the period of incubation is usually young. It can be from 5 to 30 years. So during 30 years, you can be completely asymptomatic, and suddenly in 6 months, you die. I mean, not you, but the person that are infected. The issue is fatal. Except miracles, but so miracles it's very rare. So all the patients unfortunately die. There is any conventional immune response. I mean, if you take the blood of someone that's infected by prions and you do serology tests, you will not see any marker that can show that this person is sick. And what's very, very important in prion pathology that it's transmissible and infectious. I mean that if I take the brain of someone and put it in the brain of make it eat it by someone else, this person will die. So the prion pathology has three main determinants. You can have a sporadic, so it means that, for example, here these values is coming from Europe, so it can be a little bit different in all over the world. So in Europe, you can have the sporadic form. It's constitute 1 or 2 cases per year per million. No, in fact, it's from France. It's one or 2 cases per year per million. It's called sporadic Creutzfeldt-Jakob. It could be infectious, typically crew pathology. That's based on cannibalism. These people develop prion disease, transmissible encephalopathy spongiform, just by eating brain or muscles of the died persons. You have the Creutzfeldt-Jakob variant, Creutzfeldt-Jakob, that in Europe it was a main concern because due to the mad cow disease. And you have also iatrogenic forms. So iatrogenic forms mainly do that you go to the hospital for a specific disease. You receive some medications that was purified from human origin, like growth hormones, and you go back home, and 10 years later, you develop prion pathology. You can have also genetics. On PRP proteins, you have more than 30 mutations that induced Creutzfeldt-Jakob. So you have the familial Creutzfeldt-Jakob. For example, in Chile, you have a typical mutations that generate a typical Creutzfeldt-Jakob disease. You have, so this name is impossible to pronounce, it's Grashman-Straussel-Schinker disease. that is transmissible. Is several mutations that induce this pathology. And you have another one that's fatal familial insomnia. And this mutation is quite interesting because they don't die from prions, but they die because they cannot sleep. They die because they are tired, in fact. So all this, also the genetics, are infectious. It means that if you take a piece of brain of these people and you put it in the brain of the normal persons, they will develop prion pathology. Just something. Don't hesitate to ask questions. As the schedule is quite short and everybody is hungry, it's good to have an interactive presentations. So concerning the molecular basis of prion disease, so prion disease belong to pathology like Alzheimer's, Parkinson, or Huntington, and also SOD, what we call protein pathology due to misfolding proteins. What's very important, that the misfolding is autocatalytic. It means that when it starts, it will never end. So you can do whatever you want. It will continue. The aggregations will go over. And more or less, it's the same with Alzheimer's and Parkinson's and also SOD. The concept of the prion was introduced also in this pathology. The misfolded form of PRPC, prion, is called PRPSC, SC for scrappy. And the normal form of the protein is PRPC. And as I told previously, the evolution of the prion pathology is always associated for this misfolding process. So we have no idea of all the steps that conducing from the normal proteins to the bad form of the proteins. But what, in fact, it is the main research area in the prion field. So a few words about prion proteins, the normal proteins, the good proteins. So when you look, this is the amino acid sequence of the protein. When you look this, it's very common proteins. Nothing can we cannot see from the sequence that, okay, these proteins could be bad and form amyloids. This protein is formed from two domains, what domains called N-terminal domain that start from here till around here, and have the particularity to be able to bind copper and some other metals ions. And you have the globular part. It's nice to it's nice. And the globular part that's able to bind nothing, in fact, but have two glycosylation sites and a disulfide bridge. One important thing, these proteins, the PRP proteins, is a cell surface membrane proteins. It's very interesting. And it contrasts almost with other pathology that's due to protein misfolding, like Huntington and Parkinson, that the amylogenic protein is intracellular. So this is some point very interesting. And only A beta that's coming out of the cells also for the aggregations. So you have a signal for anchoring the proteins on the cell surface. It's not a transmembrane proteins. It's just extracellular membrane proteins. This is the crystallographic structures of the PRP proteins that we resolved in 2006, with the N-terminal part completely unfolded and the globular part mainly formed from three alpha helix and a short two short beta sheets. The physiology of PRP proteins. So PRP proteins, the normal one is an ubiquitous proteins, is mainly expressed in the brain, in the central nervous system, but you can more or less find it all over the body. One important thing is that the PRP protein is highly conserved. The difference between human and the mice, for example, is only 5 amino acids residues, but it's very conserved. If you build knockout mice, that's quite easy to do, the mice are very good. No apparent physiology difference in with the wild type. It means physiological, but okay, in terms of cognitive, nobody talked with the mice, and so make a psychoanalysis of the mice to see if everything is okay. but so they are okay. Concerning the function of PRPC, in fact, we have not so much idea about its function. However, PRPC is proposed to be involved in several regulations pathway. It was proposed to be involved in metal homeostasis, to stress control, to transporter of metals, SOD activity, anti-apoptotic activity, receptor for peptide of A-beta. So a lot of activity have been proposed for PRPC futurity. So in fact, we have to say we have not so much idea about his real functions. That does not mean that it's not be involved with all of this, but it seems that it has not have a specific role. Concerning the PRPSC protein, so the bad one. So when the bad one is generated, we have a change of its structures. Its content of beta sheet increase. It become resistant to proteases. It become insoluble. And here, it's typical aggregates extracted from brain of its sheep one, at least, from a sheep infected by scrappy. The structure of the bad one, unfortunately, we don't have the atomic resolution structures. In fact, because more of the attempts to resolve the structure of these proteins, the bad proteins, completely fail. The main reason of this is not because the scientists are really incapable to do it. It's because the protein is highly heterodispersed. So most of the technique to do the structures are done to deal with very homogeneous structurations, and so the conventional techniques fail to be applied. Typically, I show you here a size distributions of the bad proteins, and as you can see, so it's highly heterodispersed in terms of size. However, there is some models, and they should be taken as models. So different models exist. One of them coming from our labs. But I have to say that all of these models fail to fit with the experimental data. So they are not all able to describe the physiology, the physiopathology of the Prion's disease. Concerning the mechanism of the conversions, I intentionally put these dates here and here. In fact, since we work on prion since, let's say, 30 years, our best knowledge about the conversion process is date from 67 or 1993. We don't have much more better knowledge on the conversion process. That's these two cartoons. The first one is coming from Griffiths, that was a mathematician. And he proposed a autocatalytic process based on the very well-known chemical reactions called Belousov-Zhabotinsky, so I will not detail in this. And the more recent, let's say, 1993, it's based on nucleation elongation process that was used to describe tubulin formations, tubulin filament formations, or flagellins formation. So this is the two schemes that we have at best. So now a few words also about the prion theory. So in fact, the prion theory should be taken very simply. In fact, you have two objects. The proteins in what we call prion states, and its substrate of conversions. This what we call this conformer, the prion state conformer, will find the good proteins, the monomer. It will interact with this through an interface of interactions and use transfer in structural informations from the blue one to the red one and convert and going so on. So in an autocatalytic process. But if we take this process like this, in fact, it's not so much sufficient to describe what we call the propagation and spreading of the the prion disease in a tissue. Why? So if this is the prions, I put it in the brain of someone. This is the particle of prions. This one will take the good proteins and convert it. So what is going to happen? It will increase in size. At one point, it will get out from the ear. This will be so long. So not so much pain, except some damage on your head. But okay, not so much efficient for the replication of a pathogen agent. So people let's say in late 90s was obliged to introduce what we call a fragmentation process to generate de novo interface and introduce the concept of amplifications. And it's why now actually we are obliged to describe the prion process by a combination of elongations and fragmentation process. Then the fragmentation process is something to introduce the concept of amplification of the pathogens. Now, how the prion concept has been demonstrated? Because, okay, at the beginning it was a theory, good theory, well-founded on terms of molecules, but was not demonstrated, let's say, till 2004. The first experiments that come and make strong the prion theory was done by when people was able to construct the wild-type knockout mice that the genes of PRPC was invalidated. So in these experiments, people take the prions inoculated to the wild type. So the wild type mice died after 150 days. And when they take it and put it in the mice without PRPC, the mice was very healthy and no significant sign. So this experiment showed that the expression of PRPC was absolutely required for the propagation of the prion pathology. The second experiment that's coming a few years later was the graph of piece of brain on the knockout mice. In these experiments, it was quite very elegant experiments for this stage of the knowledge on prions. People graph a numerous piece of brain on the knockout mice and inoculate in opposite directions prions. And what they observed is that you have replication of prions with all the histological sign only on the graph of mice that are expressing the PRPC proteins. So this is also one of the main important validations of the prion hypothesis. And one thing that I think that this is one of the more solid evidence is coming from the mutations. So we are able to generate transgenic mice with some specific mutations that make that PRP change conformation spontaneously and form prions. So this kind of mice has been built and generate, and when they develop the pathology, their brain was make homogenate and inoculated on wild-type mice, and the wild-type mice developed the pathology. So this was one of the main clue of the acceptance of the prion hypothesis, and finally, the final evidence of prion hypothesis was when people was able to produce proteins from in E. coli, PRP proteins in E. coli, and use its aggregations in fibers and put it what we call synthetic prions in transgenic mice, and these mice develop pathology exactly like they receive prions coming from the infected brains. So this was the final proof of prion hypothesis. So another main concept in prion disease is the existence of what we call prion strains. So this is quite hard to understand. I will make it a little bit simplified to make it clear. So in fact, we don't have just one kind of amyloids or prion aggregates in the brain. We can have several one. And this broad panel of prion aggregates have different biological activities. That's what we call, when they are expressed, strain phenomenon. So let's take blue fibers and red fibers. The amino acid sequence are exactly the same. It's just the conformation of these aggregates that change. You take the blue aggregates, you put it in these mice, and these mice develop pathology, let's say, in 300 days, and with some specific histology called pattern. You took the red fibers, the isolate B, you introduce it in the mice, and what you observe that, okay, these mice will develop, let's say, in 150 days, and with another pattern of physiopathology. And this difference of pattern of physiopathology is only dictated by the conformation of what you injected in the brain. And this, we use it as to say, okay, we have a strains phenomenon, and strains are completely associated to the structure of these assemblies. Here, typically, I show you just for the same amino acid sequence, SHIP PRP, you have more than 40 different strains. It means that 40 different phenotype of the disease. Some of them can pass to human, some of them cannot pass to the human, and you have what we call a species barrier. So, before going further, I make a synopsis of what I said previously. So, the prion phenomenon is an autocatalytic process of the transmission of structural informations, what we call templating phenomenon. And for the spreading and amplifications, we have what we call fragmentations. So we have templating associated to fragmentations that make prions being a pathologic agent. Yes, of course.
I was wondering if you have the same sequence of amino acids, can you interchange from one structure to another, or is it fixed once the structure is
No, yes, it's really good questions. Yeah, I catch your main. It means that, if, for example, if I take this, the blue one, I can propagate it on a mice, let's say, for 40 years. I mean, a serial passage. It will always stay the same, what we call cloning strains. You see? It will not switch on A to B. Once you fix a strains, it's going to remain these strains. This is only due to this information is only encoded in the structures of the assemblies. Okay. Now I will present you some new data that we get about the structures of this the prion replicative elements. So one of the main questions in prion field is that, okay, we have fibers, we have assemblies, we have aggregates. I don't like to use the name of fibers because, in fact, we have fibers when we purify the assemblies. In the brain, we have aggregates, so we have to talk about aggregates. We have aggregates, but what in these aggregates harbor what the replicative informations? What's the element of that's in which the strain information is encoded in prions during the replications? So here you have typically the aggregates purified from brain of hamsters infected by strains that we call RML. And as you can see, okay, we can see a little bit fibers organizations, but more or less it's aggregates. And here you have a pattern of size distributions as was evaluated by analytical centrifugations. Here you have the normal PRPC, and here you have the aggregates. So normal, it's monomeric, and it's on top of the gradient, when the aggregates are segmented all along the centrifugations tubes. So to go and determine what's the support of the prion informations, we do very simple approaches, almost archaics. And what we take, we take brain homogenates from hamsters infected with scrappy strains. We do what we call unfolding process by treat them by chaotropic agents. And one of the chaotropic agents that is commonly used to explore the unfolding of proteins is urea or guanidines. So we treat these assemblies by chaotropic treatments. We analyze their size distributions. We remove these chaotropic agents by doing dialysis or whatever. That makes that we have a refolding step, and we analyzed the size distributions and the infectivity measurement of these two treated objects. And here, what you have when you treat these assemblies with a chaotropic, you go from broad distributions of object, and you make them become small because you use the unfolding process. And here you have a typical unfolding and depolymerization process from large object to a very small object. And this can be modelized by at least two different processes. PRP-SC that's go and unfold and form an unfolded state of PRP, or PRP-SC go and unfold and generate a small object that co-sediments with PRP-C at one point. If I'm not clear, you stop me and we can go a little bit in more detail now. So when we take these assemblies and we try to remove the urea and make them refold, what we observe that so we go from there, we generate small object, we use a refolding process, and what we observe is the formation of large aggregates. From small aggregates, we go back again to the large aggregates, and what we call RF-PRP, for refold PRP. And this refold PRP was quite unexpected, because when you unfold aggregates proteins, what you are expecting that you generate just monomer and nothing else. And when you remove the chaotropic agents, they should stay also monomeric. But here in this process, we generate an aggregates once again. And our concern was, okay, we cannot have an unfolded states here, and what we have is something that's go and form something that should have probably some activity. And one of our concern was, okay, if this object have a same activity, biological activity, and infectivity that what we have originally. So for this, we take this object, the RF1, and we inoculate it to mice, and we compare it with the original Prion strains. And what we observe is that where the refolded object has exactly the same strains pattern that the original one. But the unfolded one remains completely inoculate for the mice. The mice remains healthy completely. So our conclusions at this stage was, okay, during the unfolding process of the PRP assemblies, we are able to generate something that's very small. This loses the infectivity, but when you remove the unfolding agents, it's recovered against the at identical the strain information of the original strains. So to go better inside the characterization of this small object, we use what we call size exclusion chromatography, and we isolate some objects that's very small, and we characterize this size, and it seems that for this prime strains, this correspond to the trimer of PRP. So it means that the information that was encoded in this that's contained the strain information was encoded in the trimer of the PRP instead of the whole assemblies of the PRP, or the whole aggregates. And here you summarize the process of unfolding. So you have the aggregates of PRPSC, you unfold, you generate something that is very small, and this is in a reversible form. Our next questions was, okay, we observed this process during the unfolding of the PRP-SC assemblies, so the Prion assemblies. So what's going to happen, in fact, in vivo without the context of unfolding process? So what we did, in fact, in these experiments, is that we take the Prion assemblies, the aggregates purified from brain of hamsters, and what we did is that we induced the dilutions of these assemblies. And what we observed a relaxation of their size, as observed by static light scatterings, that leads us to measure the variation of the molecular weight of the assemblies in a very small assemblies. So it means that the aggregates of prion that was normally accepted to be very deadpan object, very static object, just by simple dilutions go and evolve to a very, very small object. So this was the first time that it was shown that prion assemblies are a very, very dynamic object. To make a parenthesis, we do the same with A-beta purified from brain of some specific transgenic mice. And if the A-beta assemblies more or less has the same behavior. So the A-beta assemblies from these transgenic mice are able also to lose a small object that can be diffused and go elsewhere.
How did they migrate this small?
Sorry?
Can you follow the small part?
In the brain, no, but in vitro we can follow them. Yes. Yes, because they are very very small. They are very small in size. It means, for example, the size for these strains, the size of the object is more or less about a trimer. It means 120 kilodaltons. So it's almost nothing. It's 2 times more than BSA proteins. So they can be very diffusive, and they can be spread by the cephalorachidinal liquids, in fact. You see. So this experiment showed that there is an constitutional equilibrium between prion assemblies and their what we call elementary subunits that can. It's okay. Oh, okay. That can go. Almost finished. And so they can diffuse and spread the prion replication center elsewhere. So to conclude, voilà. To conclude, so as I told, prion assemblies are a highly dynamic object. Recently, for the first time, we are able to obtain by atomic force microscopy in liquids and from purified assemblies from, this time not from hamsters, but from Creutzfeldt-Jakob disease transmitted to human transgenic mice. These elementary subunits that you can see here. And the existence of such elementary subunits, a small object, it's very challenging in prions field because, in fact, everybody thought that the propagation of prions is just only addition of the monomer on the extremity of the existent fibers. But in fact, it's not so working like this. It's a little bit more complicated. And also concerning the fragmentations. In fact, as you have an equilibrium between PRP assemblies and their elementary subunits that keep the information, replicative informations, this object can diffuse and spread without the contribution of any fragmentation process. So this point is very, very important. And now we are revisiting the concept of the fragmentations for the spreading of prion disease. And I would like to finish with this, just to show you that this is an experiment of what we call templating process, prion aggregates with monomeric PRPC. We are expected that we have something like this, just elongations and depolymerizations. But as you can see here, this is a kinetics as was followed. Things are very much complicated, much complicated. At one point, we have oscillations of the size. This is completely unusual. That make that the systems is very, very far from the equilibrium. And such kind of system that's far from the equilibrium means that we have a networks of exchange between different subassemblies, and in a network you can store informations. So this point is very important. And to finish, I would like to thank all the people that contribute to this, and all of them are not represented here, and my groups, our collaborators, and also a very strong collaboration with mathematicians that we have, because a big part of the Prion's studies is dynamic of assemblies, fragmentation, depolymerization, kinetic studies, and for this we need a contribution of mathematicians. Thank you very much for your attention.
If there's any question, we have time for a few questions. If you have, yes.
I have one. If I get the brain from a person with one of this kind of diseases,
Yeah.
I will develop the same disease, or do I have something from
No.
myself that can modulate the
Okay.
course of the disease?
A very good question, and it's more or less joined the question of Monica. In fact, if, for example, I have Prion disease. I have Prion disease. It doesn't work, in fact. Oh, you can.
It's working.
Okay, it's working.
Not there.
Okay.
It's for the streaming.
If I have Prion disease and I'm dead, you take my brain, you make it. No, don't eat it for the moment. Just study it for the moment. You make histological studies. You're gonna see that I have a deposition in hippocampal, not on the cortex. Different characterizations. Now you take this my piece of my brain, you eat it. You are gonna develop the pathology. Someone else gonna do exactly on you, and you will have exactly the same. Exactly the same. Let me tell you, when we do serial passage of strains, the mice should die at 125 days. Okay, we can have 2 days variations, but that's it. They die at 150 plus or minus 2 days. Normally, we are ready in animal facility. Okay, normally this week we are expecting to have 20 mice that are gonna die. So we prepare ourselves. You see?
Yeah.
So in some case, you can have modulations. And this is very important, and this modulation is what we call mutation and evolution of the prions assemblies. And in which case it's happier. If I take prions from mice, a mice adapted prion, now I inoculated to, let's say, Carola. Carola will not get ill. Healthy, nothing. When she will die, as late as possible. I take her brain, I make histology, and I will do some characterization, a presence of PRPSC, such kind of things. She will be full of PRPSC, but no disease, no lesions. Now I will put it in another person, another human. The human will become ill. Let's say at 50 years old. This person, I will take it, I will do it on another human, 20 years. And the pattern evolved till stabilize. And this is the adaptations.
Okay. Yes, but you have to remind everybody that you're talking about the protein, so mutation doesn't mean...
Yeah, no, what we call structural mutations. It's very important.
Yes.
Yeah, structural mutation and evaluations. And it's why such kind of behavior, because it's important, because in what we call, in terms of physics, it's very important. In systems that are out of the equilibrium, you can encode a lot of informations.
If the inoculation happens on different stage of development, do you get different...
Yes, of course. Such kind of things has been done, not by our labs, but has been done at the beginning of the prions. In fact, before the theory of prions, it was done in late 70s, and more or less the same. The development age doesn't matter.
Ya, para finalizar, ¿alguien más tiene? Sí, you have a question.
It's a simple question. What is the function of the normal prion protein? It is known. What is the function?
No, this is there is a lot of spectra of functions, transductions of apoptotic informations, binding of A beta receptor of A beta, oligomers, receptor of two others proteins, LRP. No, there is a lot of SOD activities, stress control, oxidative stress control, a lot of things. But it seems that there is not there is a canvas of activities, but not a specific activities. This is one point. This is one important point.
Yes. Sí, sí.
No habla español, but I think that they...
Yeah, I can translate.
Respecto a eso mismo, como si que se vio algún fenotipo interesante de los ratones knockout para PRP.
If knockout mice have a particular phenotype that you can actually...
Okay, I said no, but in fact, depending on knockout, each lab has his own knockout, in fact. You understand? Okay. Some of the knockout had sleepless problem, but it seems more linked to the transgenic process than to the deprivations of PRPC. Something that you should know, that when you do a transgenic mice and you remove a proteins, even if this proteins is very, very important, as soon as you get animals, it means that you have a compensatory effect. Yeah. There is a lot of PRPC-like, yeah. There's at least two others, Doppel and shadow proteins. That's more or less what we call PRPC-like. There is another question, I think.
No more questions? Yes?
About the knockout mice, there are multiple knockout mice with a PRPC-like proteins?
What do you mean multiple?
Multiple knockouts with PRPC-like one.
Yes. There is. And it's very interesting. One of them are completely sterile. Yeah. It means that one of the, if you have PRPC, they are not sterile. But if you deplete, you remove PRPC and shadow, that's a ortholog of PRPC, atrophy of testes, and they don't have...
Actually, I work in a lab where they had a knockout mice that it was quite particular, and when we were mating them, we have few pups, mice, newborns, and then after they were mating less than normal mice. So maybe there's something there, but we actually don't.
Just to finish, because I will stop after this. We talk about A beta, we talk about SOD, we don't talk about alpha-synucleins, I think.
No.
Okay. The concept of prions.
Tomorrow maybe, because tomorrow it's Parkinson.
Okay, so probably you will hear. It seems that the prion concept was introduced to all what we call amyloidogenesis disease. So more or less, I'm not saying that they will be more exactly the same process, but some of the autocatalytic replicative process seems to be found in this also Alzheimer's, Parkinson, and SOD disease. Yes. So this point is quite important.
Yes, so I will ask Gustavo, yeah, to give Dr. Uman Rezaei, I don't know. Thank you very much for your collaboration to the course, and we see each other.